Good afternoon, everyone. Thank you so much for joining us for this webinar on what it means for human flourishing, CRISPR. If that's what you're hoping to be with, you're at the right place. So my name is Peter Notebaum. I'm with the Canadian Council of Churches, and happy to just say a few words to get us started here about the Council and about the Faith and Life Sciences Reference Group. I first want to mention that it's Holy Week, Wednesday of this week for churches in the Western tradition. And so just want to acknowledge that many folks are in the middle of religious holidays. And so thank you for taking time to join us. And just want to acknowledge all that that means for so many people who are with us today and looking forward to reliving that story this weekend. I also want to acknowledge, of course, that the council has churches in nearly every community across Canada. And so the Canadian Council of Churches and its member churches are always on First Nations, Inuit, Métis, and First Nations territories. And so we want to first acknowledge that too, that we are in that sense guests on the land here. So first a word about the council. We were established in 1944, and our mission is to respond to Christ's call for unity and peace, to seek Christ's truth with an affection for diversity. That one's especially important for our session today. And to act in love through prayer, dialogue, and witness to the gospel. And in our membership, 26 member churches, there are Anglican Lutheran churches, Evangelical and Free Church churches, Eastern and Roman Catholic churches, historic Protestant churches, and Eastern and Oriental Orthodox churches. So there's a broad set of Christian traditions who are part of the council through their membership here, and we're grateful for their ongoing support and for their guidance and leadership. The governing board of the council established the Biotechnology Reference Group already in 1999. So this group, now called the Faith and Life Sciences Reference Group, has been going for 22 years or so. And I remember early on, that was just when I began working at the council. I remember early on that one of the remarks that struck me at the time was that technology time is ticking along so quickly, and scientific time is kind of struggling to keep up with all the changes in technology, but moral, theological, ethical time is even slower. And so, that's always been a theme for me in the work of the Faith and Life Sciences Reference Group, of trying to offer a helpful critique, a useful lens, theological ethical guidelines for people of faith and scientists who are doing this work, to try to keep up with all these developments. And today's session on CRISPR is another example of that. So over the years, the Faith and Life Sciences Reference Group has been on the cutting edge of many different things. Supreme Court case on the ownership of life, who owns the progeny of mice that have been genetically modified to produce a particular cancer gene. That was called the Harvard OncoMouse case. This group was involved in that already some 20 years ago, frequently reflected on questions related to the sacredness of life, faith and human genetics, guidelines for theological and ethical reflection, technology in the image of God, artificial intelligence. These are the kinds of topics that this group has been working on very doggedly and persistently for many years. And I think they're a real gift to the ecumenical movement in Canada, to communities in Canada and globally too, for that matter. So this is just a brief word of introduction from me. You may want to just introduce yourself on the chat box. Some of you no doubt know each other. I'm so grateful to see friends and colleagues and others who I don't know as participants here, but feel free in the chat box to just introduce yourself. You might want to mention what church or community you're connected to, or what traditional territory you're joining us from. Please feel free to put that right into the chat box, and so introduce yourself to the others who are here. We do want this to be not just a presentation of information, but also an engagement with one another. I also want to invite you, if indeed you're so moved, to consider making a charitable contribution to the Canadian Council of Churches to contribute to the work of the Faith and Life Sciences Reference Group, and a link to that Canada Helps donation page will also show up now in the chat box. All right, the Faith and Life Sciences Reference Group is led by two incredibly powerful co-chairs, Dr. Moira McQueen from the Canadian Catholic Bioethics Institute, and the Reverend Dr. Peter Kuhnert, who's a practicing physician in Kitchener-Waterloo area. It's Peter Kuhnert who is moderating or hosting the conversation today, and he'll also introduce our speaker. So without further ado, and with that basic introduction, and so nice to see you all introducing yourselves to one another in the chat box. Thank you for that. Let me hand it over here to Peter. Over to you, Peter.
Thank you, Peter. Good afternoon, everyone. We are thrilled that you have been able to join us for this first of our two-part webinar series on CRISPR, today in particular looking at how that affects human flourishing. A couple of housekeeping items before we proceed. First, if you are having any technical issues with the webinar, please private message either Nicole Rokas or Rebecca Ivanov in the Zoom, please, if you're having any technical issues. Second, we will be taking questions after our keynote presentation. Please type your questions as they come up in the chat, and we will be reading the questions from the chat after the presentation. There will be some resources for further reading for those of you who may be interested after this presentation. In advance of our second webinar at the end of April, April 28th, there will also be a brief exit survey for attendees so that you can help us polish these presentations for future webinars. Allow me now to introduce our keynote speaker today, Dr. James Rusthoven, who is a member of our Faith and Life Sciences Reference Group. Dr. Rusthoven was born and raised in the Chicago area, attending Trinity Christian College and the Abraham Lincoln School of Medicine at the University of Illinois. He came to Canada in 1975 and did postgraduate and medical residency in Toronto. This was followed by a fellowship in infectious diseases at the University of Chicago, including research in immunology at the Ontario Cancer Institute. He completed his formal medical training with a fellowship in medical oncology at the Princess Margaret Hospital in Toronto, and became board certified in medical oncology, both in Canada and the US, as well as in infectious diseases in the United States. Dr. Rusthoven has practiced medical oncology and taught medical trainees while doing immunologic research at the Toronto Bayview Regional Cancer Center and the University of Toronto for 6 years, followed by 10 years of practice, teaching, and clinical research at the Hamilton Regional Cancer Center and McMaster University. While continuing to teach medical students and residents, he worked in clinical research for Eli Lilly and Company for 3 years, co-leading the clinical development of a drug, pemetrexed, for the treatment of mesothelioma, a form of lung cancer. He was then appointed consultant for the Cancer Center to the Oncology Clinic at the Brantford General Hospital, where he practiced part-time while completing graduate work, first in biomedical ethics at the University of Toronto, and subsequently in theology at Trinity College, University of Bristol. He taught biomedical ethics part-time in several institutions and became a professor emeritus in the Department of Oncology at McMaster in 2014. Dr. Rusthoven finished his medical career as a medical officer and medical evaluator of new cancer biologics for Health Canada from 2016 to 2020. Dr. Rusthoven is an active member of the Christian Reformed Church, and as I mentioned, a member of our Faith and Life Sciences Reference Group here at the Canadian Council of Churches. When not engaged in family or church activities, he enjoys bird watching, photography, hiking, and bicycling. It is our great privilege and honor to welcome you, Dr. Rusthoven.
Well, thank you very, very much, Peter. And Peter, let me just get my act together here. How's that? Does that work? Okay, well, we're here today to talk about CRISPR. We're here to talk about antiviral to going viral, which means it is a natural part of the bacterial immune system against viruses, but it's also going viral in current terminology in terms of its rapidity of spread and development. I have no financial conflicts of interest to declare. I will, however, declare my biases. I'm a reformational Christian in terms of my worldview. I believe that human flourishing is fulfilled through faith in Jesus Christ. And I believe that Christians should promote biotechnology that could care for the welfare of human beings and the world around us with acceptable risks. I'd like to also draw your attention to a group resource that we have at the Canadian Council of Churches. It's a faith and genetics curriculum, which was developed a number of years ago. It has everything from basic genetics, including the genetics of sickle cell disease, which we'll talk about a bit later in this talk, as well as sessions on faith and human dignity, faith and social justice, etc. So for your future reading, perhaps before you attend the second part of these webinars. So let's first start at the beginning. I can't assume anything in terms of the participants' knowledge of genetics. So let's just talk a bit about what the difference between RNA and DNA. The DNA, of course, is a double helix discovered in the 1950s. It has a sugar phosphate backbone. If you unravel it, it looks a bit like a ladder. You've got one strand running this way, one strand running this way, and in between, you've got these nitrogenous bases, 4 of them, which are always linked in a specific set of pairs, adenine and thymine, guanine and cytosine, for example. These are linked in this ladder by hydrogen bonds, which individually are very weak, but together form a very stable and in conjunction with the double helix, a very lasting biological entity. DNA carries the code for forming proteins, which are important and necessary for our cells' existence and function, as well as our function generally. RNA transcribes the code on DNA. You can see this strip of RNA, and RNA is normally as a single strand. This represents this strand on the DNA, as you can see from the matching code. The difference is that you've got uracil in RNA as opposed to thymine in DNA, and that's only difference by a simple methyl group. So they're very similar, and RNA takes the message of DNA out of the nucleus into various parts of the cytoplasm where proteins are produced. So that's the basis of what we know about genetics, as well as what CRISPR does. So where is CRISPR found in nature, and how can it become a tool for gene processing? Well, it's been about 25 years since a curious group of sequences was discovered by a Japanese investigator. And these were base sequence repeats in the DNA of E. coli. Now, over the next 15 years or so, these were repeatedly found in different bacteria, and someone came up with the phrase clustered regularly interspaced short palindromic repeats, or CRISPR as we know it now. A few years later, the Spanish investigator Marica came to the realization that the intervening sequences or spaces between the regularly repeated parts in the bacterial DNA were actually derived from foreign genetic elements, not the bacteria's own DNA. 3 years later, the pieces were again being put together so that it was realized that CRISPR had an RNA guide, which was connected with an endonuclease. And this is an enzyme which can split or cut DNA or RNA, and its short is Cas, so CRISPR associated for Cas endonucleases. The gene for Cas was discovered a few years later, and in 2011, Emmanuel Charpentier in France actually discovered what's called a transactivating encoded RNA molecule in the bacterium Streptococcus pyogenes. And the importance of this was a key element of what we learned later to be CRISPR in activating the cutting mechanism from the RNA guide. She got together with Jennifer Doudna, who was at the University of California at Berkeley, and within a year they had completed the CRISPR system and first reported it in Science in 2012. They concluded at the end of their paper that zinc-fingered nucleases and transcription activator-like effector nucleases, which were predecessors of CRISPR, have attracted considerable interest as artificial enzymes engineered to manipulate genomes. We propose an alternative methodology based on RNA-programmed Cas9, which could offer considerable potential for gene targeting and genome editing applications. The key to this is the RNA-programmed Cas9. These nuclease systems do not have an RNA guidance system, which makes a big difference in both the efficiency and precision of the methodology. 8 years later, they received together the Nobel Prize for Chemistry for this discovery and its application from bacteria into the laboratory and for gene editing in various areas. In 2018, so much was being published about CRISPR, it warranted its own journal. So if you have a few moments, you can have a look at what CRISPR Journal has to say. I won't dwell on it today, but it is a very interesting journal. And I point to the logo here, which is quite cute. This is a picture of the RNA guide, which is part of a scissor, which is the splicing mechanism for CRISPR-Cas. So a little bit of aesthetics there as well. So again, CRISPR, which stands for clustered repeatedly interspaced short palindromic repeats, are a normal bacterial innate immune system. It's found in about 30% of bacteria and about 90% of what are called archaea, which are similar to bacteria, but exist in remote areas in very extreme conditions. There are short segments of DNA. There are spacers of DNA between those palindromic segments. And there's also nearby a gene for the Cas proteins, which are the splitting or cutting part of the complex. So here you see diagrammatically a bacterial genome, which has the spacer DNA, which contains bits of virus, which had invaded the bacterium previously, and subsequently was stored from memory, like we would have in memory cells in our spleen, for example. Well, what's the palindromic part? Well, if you look at a strand of DNA, as I mentioned earlier, one strand runs in one direction, one strand runs in the other direction in terms of these sequences. They're normally not in any particular pattern. In a palindromic sequence, however, the strand running this way will be identical on the other side of the strand running this way, so that, in fact, the code is exactly the same, just running in reverse directions. What importance does it have? Well, it's certainly a signature for CRISPR, if you see it in bacteria, but it has no other major function as we know it right now. So what do bacteria do? Well, a virus comes along to the bacterial cell membrane. It spits out some of its DNA or RNA. Some viruses are RNA viruses. The bacterium recognizes this. It takes a piece of this, sticks it in a spacer between these palindromic repeats. Over time, you get what's called a CRISPR array, which is a number of different spacers which have different segments of virus and essentially forms a memory bank for the bacterium. Nearby are the Cas protein nuclease genes. If that virus or a very similar virus genetically comes along, the spacer will produce a guide RNA from the DNA. It will link up with a protein produced by the Cas genes, which will be a nuclease. These together will roam through the cytoplasm, find the intruder, and chop it up, essentially. So this is the best diagram I could find of the complex of CRISPR-Cas9. Cas9 is just one of a whole group of different kinds of splicers, which we won't talk about today. But essentially what happens is you have a double strand of DNA like you see here. This little bulge in the middle. You've got the RNA guide, which is composed of a spacer or a CRISPR RNA segment, which is the code that recognizes an area of interest on the DNA of interest. You then have this thing that Emmanuel Charpentier discovered, which is an activating portion, which then activates the CRISPR and allows it to cut both strands of the DNA. There's also another part here, which I won't go into, which is essential in the guide finding the appropriate place on the DNA as instructed by its producer. So this is really the nutshell, the origins of CRISPR. It's the kind of CRISPR that was used about 2 or 3 years ago. There are now newer forms, which we'll talk about in a moment. Well, once you cut something, usually you also need to fix it. And the human body has 5 or more different ways of repairing broken DNA, which usually occurs when a cell divides. In terms of CRISPR, the 3 most important ones are base editing repair, where you've chipped out just one base pair, just one letter, as it were, in the code. And that's true in the case of sickle cell disease. More generally, you've got the non-homologous end joining mechanism and the homologous directed repair mechanism. This is a bit of a dirty mechanism. It just sort of patches the spliced ends and often produces errors. With the homology directed repair, you've got a much better patch, which actually better represents the DNA that you want put in that defect if you're trying to do an edit. You may have heard of BRCA1 and BRCA2, which are breast cancer mutations. They are actually mutations that cause defects in this repair mechanism in human beings and is associated with a very high incident risk of breast cancer, ovarian cancer, and other cancers. So these repair mechanisms are extremely important for all sorts of reasons. And if you don't have a major one, you could have serious problems in the future. So again, you've got the CRISPR mechanism here, doing a double clip on a segment of DNA. That repair can either be done by the non-homologous method, putting in an indel, which stands for either an insertion or a deletion of a small piece of DNA, or it can use this method where it actually takes some donor DNA that was introduced into the environment and actually put that as a patch in the defect. The opposite side will then be repaired as well by the body's own mechanisms. So right now, specificity and efficiency are 2 key words in CRISPR development as a biotechnology. You've got the guide template, which is a major improvement over the previous methods, as I mentioned before. It also can, however, cause some off-targeting errors because of similarities to the guide template. You can also take the Cas9 cutting enzyme and mutate it to form what is cleverly known as a nickase. Literally, it makes a single nick or cut in one rather than 2 strands of the DNA. And if you do that on both strands and manipulate it in a certain way, you can actually increase the specificity and efficiency considerably. So in the last year or so, there's been a new method called prime editing, which takes one of these nickases, it fuses it with another enzyme called reverse transcriptase. Reverse transcriptase converts RNA into DNA rather than the usual normal mechanism of DNA being transcribed by RNA. It also has a much longer than usual RNA guide, and that whole complex becomes known as the prime editor guide. And the reason this is really helpful is that it not only specifies the target area, but it also encodes the desired edit. So you can take along right on the guide the bit that you want to be inserted in, and using the reverse transcriptase, you can convert the RNA into a new patch of DNA right at the site. So this is all done to result in a very stable edit of DNA. On the opposite strand, you can put another nickase on the other side with a regular guide. And then what will happen is the body will use that edit that you put on the other side as a template for replicating that new side. And finally, as I mentioned earlier, single base repairs can also be done with CRISPR-Cas9, but you need another enzyme called the deaminase enzyme, and that can convert one of the base pairs, such as cytosine, into thymidine. And now we have enough of these enzymes that all 4 can be converted to other ones as directed by the investigator. So here's a more of a cartoon to illustrate this prime editor. You've got the nickase domain here. You've got the reverse transcriptase domain. And then you've got this very long RNA guide. That RNA guide searches out and finds its complement on DNA. But rather than just dwelling on this one strand, the other end actually can wrap around. It forms what's called a primer binding site. And that allows, after the clip is made here, it actually allows the reverse transcriptase template to produce a new piece of DNA that fits exactly into the deficit here. Okay, everybody take a deep breath. That's enough for anybody in terms of the basic science. So what are examples of how CRISPR-Cas9 is now being used? Well, it's being used all over the place. It has its own journal, but plenty of other journals like Nature and Science are publishing regularly at involved in gene editing in animals and crops, where the other nucleases, the talons and the ZFNs are still being used, as mentioned earlier. It's also creating models for disease where we can figure out how we can treat diseases before they're actually given to humans. Most of the technology is focusing on somatic cells. Those are cells that are not transmitted to the next generation, but it's also now being used in germline cells. And what I will do is give you a couple of examples where it's being used for somatic mutations causing disease, and one example of how the germline cells are using it. So 3 months ago, the New England Journal published 2 articles on how Cas9 gene editing was used to help patients with sickle cell disease and beta thalassemia. We don't have time to concentrate on this one, so today we'll just talk about how they did it in patients with sickle cell disease. So 101 on what sickle cell disease is. Normal adult hemoglobin has 4 chains, 2 alpha and 2 beta chains, which are proteins. In sickle cell disease, there's a point mutation, a single base, either the A or the T or the C or the G, on the beta chain of the gene. So that chain of protein that's developed has just one amino acid that's been replaced, and that causes the ravages of sickle cell disease. 300,000 individuals every year worldwide develop the disease. So the prevalence is very high. The mutation causes reduced or absent beta globin synthesis, with the resultant ineffective red blood cell development, poor red blood cell functioning, which is carrying oxygen, of course, to your cells, and anemia. So even more importantly, though, clinically, it causes an abnormal deformation or sickling of what are otherwise rounded and biconcave cells, red cells. And this causes clumping of the red cells in small capillaries in different organs. It causes oxygen deprivation because of the blockage, and eventually end organ damage and reduced life expectancy. So very, very serious disease. Now, normally newborns have fetal or hemoglobin F, which has 2 alpha chain globins, but also, instead of the beta globin, gamma globin. In the first year of life, there's a gene called BCL11A, which represses the gamma globin synthesis. So it wants the body to get rid of eventually the fetal hemoglobin and replace it with adult hemoglobin. So it decreases fetal hemoglobin, but at the same time, other genes are increasing beta globin to generate an increasing amount of adult hemoglobin. So there's this balance in the first year or 2 of life where fetal hemoglobin goes and adult hemoglobin comes because of this exchange of gamma globulin for beta globulin. But in sickle cell disease, there are low levels of ineffective adult hemoglobin A that are produced during this period, while at the same time, the fetal hemoglobin, which is normal, decreases. And this can carry oxygen quite effectively. So this causes a debilitating anemia requiring regular transfusions for patients. And this has complications themselves, including iron deposition, excess iron deposition in organs like the kidney, the spleen, and the liver. Now, what these authors did was they took the CRISPR-Cas9 system and introduced it into cells, which I'll mention in a moment, by a process called electroporation. Now, keep that in mind because that's important. And it is meant to genetically edit the region of that BCL11A gene, that's the gene that normally represses gamma globulin synthesis, with a mutation that overcomes the normal repressive effect. So essentially, you want, or the investigators want, fetal hemoglobin to hang around because, in fact, the adult hemoglobin is not going to work because of the mutation. Excuse me. And this is schematically. This is the CRISPR complex, as I've shown before, the guide RNA, the cutting mechanism, latched on to the specific area of DNA, which in this case is the gene or portion of the gene for BCL11A. And that focuses particularly on what's called the erythroid enhancer region and does a splicing of the mutation and other splicing to actually silence the silencer. So it gets rid of the inhibitory effect. So the method was they took what are called autologous, that person's own, hematopoietic stem cells, which they get from the bone marrow. And these are precursors to white cells, blood cells, and platelets rather, and red blood cells. And through the process of electroporation, which puts a small current into these stem cells, allows the cell to take up the CRISPR-Cas9 complex that can then dissect out the enhancer part of the BCL11A gene. After the editing, these stem cells are infused back into patients with sickle cell disease. And according to this report in the New England Journal 3 months ago, the earliest patient had been followed for about 16 months. Now, I want you to note that this is a method that was used here. There's another method using what's called a lentivirus vector, which was used in the other article. We'll talk briefly, time permitting, about why these are important differences. Anyway, to date, in that report 3 months ago, 80% of the sites for that particular gene were edited appropriately and maintained. Fetal hemoglobin, as a result, was increased by 20 to 40% in edited stem cells and early red blood cell precursor cells. Now, this was all done before they injected the cells back into the patient. There was no evidence of off-target editing. Again, as I mentioned earlier, this is editing that's not desired and is done at sites of the DNA that are distant from the sites of interest, but they happen as errors. But they found none after editing and before reinfusion into the patient. What they didn't do, however, was following up by testing for off-target editing later. And this is a major issue that needs to be done. The longest followed patient became transfusion independent. Her hemoglobin rose from 7.2 to 12, which is now in the normal range at 15 months. Now, it's a new biotechnology, and it has issues. Bluebird Bio is a biotech company, which is also trying to help patients with sickle cell disease and beta thalassemia. But their method is to take the mutated globin gene and replace it with a normal functioning globin gene. So a very different strategy. However, during their early phase trial, which was begun in 2017 or 18, it was stopped a few months ago because one patient developed acute myelogenous leukemia and the other developed what's called myeloplastic syndrome, which is a potential precursor to cancer. The normal gene, that is this normally functioning beta globin gene, was introduced into patients' red blood cell precursors or stem cells using a lentivirus vector. So electroporation was not used, but a virus was used which packaged the DNA. And these viruses are modified from what I call oncogenic potential. They could cause cancer when they were left in their original state, but they were modified 3 different generations and felt to be rendered no longer capable of replicating or linking up with cancer. There were no cases of blood cancers that were found in patients with beta thalassemia given the gene therapy. This was a trial in Europe. And in fact, the European Medicines Agency, the European equivalent to the FDA, had granted conditional approval for the therapy for beta thalassemia in 2019, but the company had to make improvements in manufacturing, and then COVID came along. And in fact, no patients have yet been treated in Europe with this methodology. So that's an example of its use currently in patients with sickle cell disease. We'll now just look at a case of germline or heritable gene editing where CRISPR is being applied. In July of 2017, Metelipov and his group in Oregon edited DNA from 1-cell human embryos using CRISPR. What they did was they took human embryos, they formed human embryos, which were combined with sperm that had a mutation associated with a medical condition called hypertrophic cardiomyopathy, which is a very small mutation, with a human egg that did not have the mutation. So the single cell embryo was heterologous for the mutation. So one of the pairs of genes had it and the other complementary did not. Those are called alleles in genetic language. A CRISPR-Cas9 complex was constructed that would cut out mutated DNA segment and replace it with a normal segment using a short DNA template, not unlike we saw with the prime method that I mentioned earlier. So they injected the single cell embryos to avoid mosaicism and off-target effects. Well, I mentioned what off-target effects are, trying to keep it precise to the target area in question and not landing and cutting other areas. Mosaicism is a form of genetics where if you, for example, gave this to embryos that were 5-cell or 10 cells in size, then some of those cells may grow and form without the mutation, while others will have it. So the embryo will be mixed in terms of its genetic representation. If you just do it in this first cell that forms the embryo, that's avoided. Now, very importantly, and we'll touch on this in the second webinar in a month, they were inspired to do this by the US National Academy of Sciences report in 2017, which they interpreted as giving approval for lab research in germline modification. Now, this remains very controversial. And again, we'll talk about that at a later time. Importantly, they claimed that there were no detectable off-target effects. So that's July 2017. We'll move up 3.5 years to October 2020. Zaccaro and his group reported on an attempt to repair a deletion mutation in another gene which caused retinitis pigmentosa, which is a cause of blindness, in heterozygous single-cell embryos using a similar CRISPR-Cas9 based construct. So they used a similar type of human embryo, single cell, using CRISPR to try to repair mutation in this particular gene to prevent this particular disease. They found that half of the Cas9 induced double-strand breaks were not repaired. And this left one allele, or one of the 2 pairs that that gene is on, pairs of chromosomes. That one allele then was missing. And it caused a loss. I can't read that with, let me change this a minute here. It's on my screen, I can't see it. Anyway, all the chromosomes, the whole chromosome, or a good portion of the chromosome itself, was actually deleted during the next cell division. So what's the importance of this? Well, huge amounts of DNA were lost, for one. The second is this loss was not detected because the detection methods saw normal companion chromosomes only. In other words, if a whole chromosome was taken out that had the mutation, you couldn't tell that in fact it was missing. And they suggest that Metolipov's observation that no off-target problems occurred may not have been correct if a whole chromosome had been lost in their methodology. So the bottom line of this was the way of actually detecting the efficiency of off-target problems has a lot to be desired still. And that even with a 3-year difference, there are more and more methods being developed so this detection can be much more precise. And it's extremely important because these off-target effects can produce all sorts of other problems, including diseases in and of themselves. If cuts are made in other areas or new bits of DNA are placed in areas other than the area of interest. So I'll give one more example here. There's a disease called Leber's congenital amaurosis. There are about 18 versions. One of them is called number 10. And each of these 18 versions has a different genetic abnormality. So in this case, it causes blindness because it's congenital. It would be in newborns. And it's due to a single mutation called the centrosomal protein of gene 290. Now, this mutation disables photoreceptor cells, but the cells are actually still alive. So we have cones and rods in our retina, and those cones and rods take light and transfer it into our brain. Well, these photoelectric cells are deficient in their ability to do that. And a phase 1 study was started in September of 2019, whereby microdroplets containing a CRISPR gene editing complex was injected under the retina to splice out the genetic defect and restore the functioning of the photoreceptor cells. And this is just a link to the actual details of the clinical trial at clinicaltrials.gov. Now, that gene therapy, they called it Edit 101, and it uses a different vector, an adeno-associated virus, which is essentially a virus that needs another virus to replicate. So it's like you have viruses that need another cell. They can't exist by themselves, but they have to enter another cell to get the hardware to replicate. The same is true here at the virus level. Here's a virus that needs another virus to replicate. So that virusy thing, which isn't a full virus, and in fact was denuded of its own viral DNA, that what there was of it, that vector was used to deliver the Cas9 complex and the specific guide RNAs that would fix the mutation into the receptor cells by subretinal injection. So what did he do? Well, this is the example of a cone. This is one cell below the retina or in the retina. The mutation in question is in it's this area where there are what are called connecting cilia, which allow for protein trafficking and carry light photons down toward the brain. When the mutation occurs, there's no protein trafficking because of the defect in that small section of a connector with the cilia. The virus takes the 2 guide RNAs along with the Cas9 splitter and is injected in the eye underneath the retina. And this is the strand of DNA with the mutation. Normally, the DNA message would be taken to RNA, and RNA would form the protein, which is defective. What the editing does with CRISPR is it takes out this segment. Sometimes it actually inverts or reverses, but it's connected here with the defect being removed. You can have full functional gene and therefore potential vision where there was no vision for that individual. And apparently it just takes full functioning of only 10% of the cones to have decent vision. So all that we've talked about right now really talk about how do we improve CRISPR precision? How do we improve efficiency and establish safety with this new technology, which has only been around now for a few years? Next time, we're going to talk about ethical and societal issues. What are problems with designing, implementing, and interpreting clinical trials using CRISPR? How do we apply CRISPR where no other effective, safe therapies are available? And at the societal level, we'll talk about multiple stakeholders who, and at what level do these stakeholders make decisions regarding how we use CRISPR gene editing. And this is particularly relevant for germline gene editing. I'll just end with some aesthetics. I think everything has an aesthetic part to it. Here's a model of DNA. If you actually look from the top straight down the core center of the DNA, it'll look like that. And if you've ever been to France and seen the rose windows in the cathedrals at Chartres or Notre Dame, I think you can appreciate there's some aesthetic pleasure in that. I have another case that I can present, but in the interest of time, I'll go back to our moderator and we'll take questions.
Thank you, Jim, for a stimulating conversation about a very complex topic, CRISPR. In the last 25 years, humanity has discovered this primordial immune system that bacteria have against viruses, and we figured out how we can apply that to modify human beings, not just to change potentially individual genetic diseases in one individual, but potentially to create changes that can be inherited from parents to children into future generations. So it is a powerful technology that we are just beginning to understand. And I think you've done a good job in explaining both what CRISPR is and some of its uses. Mindful of the time, we have perhaps 5 minutes for questions. If there are 2 or 3 questions that the group would like to ask of our speaker, now would be the time. And then I will encourage people to complete the exit surveys, and we'll have some resources for people to review prior to our next webinar on April 28th. Some concerns about applying the technology in humans, Jim, while we're waiting for a first question. There's a case going back to 1999 of a 18-year-old boy in Philadelphia, Jesse Gelsinger, who was involved in a gene therapy trial for genetic disease. And he actually had, in the midst of that trial, an overwhelming reaction to the virus that was bringing the gene therapy, and he actually passed away. So these therapies can be dangerous. These are not regular medical treatments. How do we handle the risks of these therapies that can cause significant health challenges for those who are partaking of them?
Well, that's part of what we're going to talk about next time at the end of April, but there's a couple of stakeholders, as I call them, that have to be involved. One is the regulators. I worked with Health Canada for 4 years, and that's not an area that I was directly involved with, but it's extremely important that regulators judge that, in fact, these treatments are not only effective, but are safe. And with off-targeting and other issues still prevalent, that's going to be very difficult. There's also differences between the regulators. We didn't always agree with the FDA, for example, in Canada. So there will be issues, I'm sure, between different regulators internationally as to when some of these can be considered safe enough to put on the market. Another issue is the patients. Many of these patients have very debilitating diseases like sickle cell disease or blindness, and they will often go to extremes to take experimental treatments which are not yet judged to be completely safe. And that's another ethical issue And I think it was partly the Gessinger issue in terms of the proper, the way that the investigator gave information to Jesse and his father. At least that's what I understand from that case. So that's extremely important that if you're an investigator and you're involved in this, that you're very clear, as clear as you can be, about the risks involved. But as I've tried to show, this is a complicated technology. It's a complicated field even before CRISPR, but CRISPR has made it even that much more complicated, partly because it is precise and it has many more components to it.
Great. Thank you, Jim. This is a question from Audrey. In your presentation, you mentioned in one of the human studies at the end of the presentation that they didn't follow up for off-target editing that CRISPR was causing. The question is from Audrey, why did they not do follow-up testing for off-target editing?
I don't know. That's another issue. If you have stem cells and they develop into red blood cells don't have DNA. They're sort of carriers of oxygen. That's all they do. But you certainly could test the precursors in the stem cells that are still in the patient and try, because we're living beings and we change and our cells change. And when you do gene editing, it's no different. You can get changes from one generation to another. And it's particularly important to understand where these vectors are putting this DNA. And we don't know. So you've got that problem, and now you're dealing with a technology that cuts DNA and puts new pieces of DNA next to other genes. And there are diseases that can be caused by doing just that without knowing that you're doing it. So you could say it's a premature release of hype. I don't think that's the case, but I think that's where the press can be very uninformative and even dangerously informative by not giving a true picture to the public when some of these come out. And I'm not blaming the media so much, but again, it's a very complicated area.
Thank you, Jim. This question is from Paul. And Paul asks, was CRISPR used to create the Pfizer and Moderna COVID mRNA vaccines?
Not to my knowledge. mRNA is included in both of those. mRNA, I believe when it's injected, goes to the lymph nodes and goes to dendritic cells, which are antigen presenting cells. But that mRNA gets into the dendritic cells when it then therefore produces the spike protein. And the spike protein is what the dendritic cell puts on its surface and displays, as it were, to the rest of the immune system. And that's where the T cells and the B cells gear up, recognizing this ain't normal, and they do their thing to try to kill an infection. But the infection, of course, isn't there. It's a vaccine. But what they do then is they take the memory of that interaction and they store it for when the real thing comes along later. And if they do it well, we've been protected.
Would it be fair to say that all the research into CRISPR, which is the mRNA technology, allowed for the development of these Pfizer and Moderna vaccines that were created in record time?
I don't think so. I think you have to be careful. Just because it says mRNA doesn't mean it has anything to do with one technology versus another that's using mRNA. I think what all of this has shown is that messenger RNA is extremely helpful and an extremely important intermediate between the code that we have in DNA and in the nuclei of our cells and the end product proteins that actually do the work for our cells. And the knowledge of that in itself is enough to generate the kind of vaccine technology that the Pfizer and Moderna products have done. But for something like CRISPR, it's a guide. RNA is used as a tracking mechanism to figure out which part of the DNA that you really want, and then it does its cutting. And that wasn't there before.
Thank you, Jim. Mindful of the time, we just have a couple of minutes left. I would like to encourage everyone to complete their exit survey, please, to tell us how you experienced this webinar. A reminder, our next webinar will be on Wednesday, April 28th, looking at who determines the course of human genome editing. That's Wednesday, April 28th, beginning at 4 o'clock. There will be a couple of links for resources you may wish to think about or read, including a booklet, Technology and the Image of God, created by our Faith and Life Science Reference Group, which is a collection of essays. As well, I would recommend to you this book by Walter Isaacson, Codebreaker. If you have time for a short 500-page read, it talks about the history of the CRISPR technology, its current clinical uses, its future hopes. Very stimulating read for those with extra time. I would like to thank Jim for leading us in this conversation today. And it's been an excellent talk, an excellent introduction to CRISPR. We look forward to the second part of the webinar series. And thank you to all the members of the Faith and Life Science Reference Group who helped make today possible. In particular, I would like to thank Grace McCreary and Nicole Rokas, Rebecca Ivanov, Joan Masterson, and Ann Mitchell, who helped make this webinar possible today. We have 2 minutes left. Perhaps there's one more question that we can deal with. One more question here. Here's a question from Nancy, a question about how does the church view pre-implantation genetic testing to prevent genetic disorders in the embryo? So one last question we can handle in this time frame, Jim. Jim, I think you're on mute.
I'd say that's a little bit off topic, and there's the church and there's the Canadian Council of Churches. So yeah, I don't think there's time to really go into that. It's a controversial method in terms of how it's used, but I don't know of any link to CRISPR on that score just yet. You're on mute, Peter.
I think we are at the end of our webinar time today. Thank you very much for making time available in Holy Week to join us to look at this technology that has the potential to change humanity for millennia going forward. We wish you all a good Holy Week journey, and we look forward to you joining us again on April 28th. Jim, a last word?
Thank you very much, Peter, and Peter Nottebohm for your introductions and for all the people who technically helped to make this possible. And thank everyone for coming to have a quick learn. And I would refer you back to the curriculum that we have to help you understand even more some of the basics of genetics, because it's here to stay, and treatments that you will be receiving and those of your children may well involve using technology such as CRISPR. And I think it behooves us all to understand that to some degree when talking to our caregivers.
Okay, thank you, everyone. Have a blessed Holy Week.
Bye-bye.